This is Neil Greenspan with Pathogens and Immunity, and I'm here today to introduce a good friend of mine who I've known about 40 years, Dr. Vishva Dixit, who's currently at Genentech. He's a world-famous immunologist and biochemist who has made major contributions recognized pretty much globally in the study of innate immunity and particular aspects related to inflammasome activation. It's a true delight to be able to speak with Vishva today and have him enlighten us on what his career in science has revealed about biology in humans, but also what it has revealed about how to be successful in science. So I'm interested, Vishva, in finding out for our audience, where did you grow up? Where did you go to school? What were the various stages in your career in biomedicine and biomedical research?
Yeah, thanks for that introduction, Neil. Yeah, I was born and brought up in Kenya in East Africa. And this was during colonial times. At that time, Kenya was a British colony. And it was another world. There was a very distinct color line and segregation of races, a sort of an apartheid system. But then independence came in 1963 with the integration of schools, and that was also at a certain level socially traumatic. But I proceeded to go through the school system there and then attended medical school at the University of Nairobi and left in 1981 to begin a residency in laboratory medicine at WashU in St. Louis.
Do you feel your experiences growing up in Kenya, and from my understanding, you were in your very early years in the more rural and isolated parts of Kenya and later more in Nairobi, the main city. Did your experiences in Kenya and different environments there, but also then in the US, influence how you think your intellectual development unfolded?
Yeah, I think I was in many ways blessed to have been exposed to such a broad spectrum of society. Certainly in Kenya, my parents, who were both physicians, they practiced in a small town in the highlands. And that was a very different existence for the Kalenjin people. And you have to realize that the people that they served were very poor. But when my father passed away in Nairobi after a long illness from pancreatic cancer, I was really surprised, shocked, in disbelief that the villagers who are really poor had collected money and paid off his hospital bill to thank him for the services he had rendered the community over the decades. So that sort of underscored to me that even though they're very different cultural settings, that at our very core, our values, aspirations are often quite similar, but unfortunately they're exaggerated and given an us versus them spin by politicians and religious preachers. And that's done as an instrument for exerting power. The other fact that I took away from experiences is that while education is key for the betterment of societies, it really has to be coupled with the respect for differences. And indeed, in many ways, diversity is to be celebrated. But I think it really, at the end of the day, suggested that we are more similar than we are different.
It's both a remarkable testament to your father and how he devoted himself to the patients that he treated, but also your comments are, although not exactly what Pathogens and Immunity normally focuses on, at least in these quarters, well received and I think quite insightful. Can you recount what early influences steered you to biomedical science in particular, as opposed to clinical practice?
Yeah, well, I was growing up in the small town, Kericho, in the highlands of Kenya. I think I was a curious kid, but my vision was seriously compromised, and that precluded any participation in sports. But I could read, and so I buried myself in books. And I was greatly enamored by the explorers of old, those who had discovered new lands and treasures. But somewhere along the way, realized that much of that had already been discovered, but that there was an equally compelling unknown to be explored in the sciences. I mean, you could be an explorer in the modern world as a scientist and have the joy of discovery. So I think that was the seed that said to me that if you want to be an explorer in the modern day world, be a scientist.
That's quite beautiful. Looking back on your, anyone would say, I think, highly successful career, can you identify one or more decisions where you think it had a major impact on what followed and that you believe facilitated your career progress?
I think it was really gaining admission to be a resident in the storied Department of Pathology at WashU. This was, of course, greatly facilitated by my brother, who was a fellow in rheumatology, and convinced the department to give me a chance and take me as a resident in lab medicine. And for me, it was just a wondrous and eye-opening experience. Not only was there an emphasis on research, but most surprisingly, at least for me, knowledge was continually questioned in journal clubs and research meetings. And presenting at these forums that we used to refer to as the Shark Tank was a daunting experience. And in hindsight, probably the experience that was most valuable to me. Because not only did you have to know your material, but you had to address a stream of penetrating questions from really astute minds in the audience. And it really made you think on your feet. And for me, it was a watershed moment. For the first time, I really understood what it takes to be a researcher, that one has to be critical of one's own work and critical of the work that you read about, critical of the work of others. So that was probably the most enlightening experience I had in my career, was being at WashU in St. Louis. It was just an amazing time.
So as you know, I was in those meetings with you.
Yeah. So you know what I'm talking about. And, sometimes I think back to them and I'm not sure that sort of tenor and that of questioning and challenging would be allowed today. At least it would be frowned upon. But I think it's a pity, because I think it's so important to be challenged and to learn from that. So that for me is the sort of crucible of knowledge that allows you to be a successful researcher.
Let's move on to a little bit about your research. What do you regard as your most significant discoveries?
I would say the first one had to do with receptor signaling. We were very interested in how death receptors like FAS and TNF receptors signaled. And at the time we embarked on our studies, receptors were thought to signal by either altering phosphorylation, dephosphorylation events, or by functioning as ion channels. But we uncovered a third way that receptors can signal. And this was through the recruitment and activation of a protease. So in other words, the second messenger was a protease, which was quite unexpected. And this is sort of common way of signaling with death receptors. So they activate these proteases termed caspases, cysteine proteases that cleave after an aspartic acid residue. But the other thing that fell out of the study of these signaling complexes was the realization that they were held together by a protein-protein interaction motif that was dubbed the death domain. And so we started looking at databases for other examples of death domains. And we discovered a lot of interesting molecules that had death domains. But I'll just highlight one of them, which was MYD88. And we discovered it had a death domain, and MYD88 was the conduit for a signal transduction from the IL-1 receptor, and later shown to be the conduit from TLR receptors. So that sort of frenzy of looking for molecules with a death domain turned out to be very fruitful. And in that search, we also uncovered the rest of the mammalian death proteases, these caspases. And so that was a very fruitful period. It happened mostly during my time at Michigan. But once I came to Genentech, this would be in 97 or so, we became more interested in the inflammasome. And so this would be a pathway that regulates inflammatory caspases such as caspase 1 and leads to the proteolytic maturation of IL-1 beta and IL-18. And during this work, we made, I think, quite an unexpected discovery. So in 2011, the Nobel Prize had been given for the LPS receptor, which was toll-like receptor 4. And one assumed that that was the way by which LPS signaled. But in 2013, we discovered that LPS could signal through a TLR4 independent manner, and in doing so, activated what we call the non-canonical inflammasome. So this was unexpected because we went on to show that at high dose LPS, TLR4 mice could be sensitized to LPS killing. So TLR4 was dispensable, but what was required was this non-canonical pathway. This, of course, has ramifications for cytokine storm and sepsis. And in studying this pathway, the non-canonical pathway of LPS responsiveness, and the approach was mostly genetic, forward genetics in mice, we inadvertently discovered how leaderless cytokines are released. So leaderless cytokines are cytokines like IL-1 alpha, IL-1 beta, IL-18, IL-33. These are very potent pro-inflammatory cytokines. And they are actually released through a membrane channel that's formed by a protein, gasdermin D. So this was a problem that cell biologists had pondered for a long time. And in the end, the solution was quite simple. When there are inflammatory insults and the inflammasome is activated, you get the creation of a pore, and this allows for the leakage of leaderless cytokines to the exterior. And then this led to our most recent discovery, that of NINJ1, which is a discovery that violates what we were taught by our high school biology teachers, because what we were taught is that following cell death, cell lysis is a passive osmotic process. But we actually found that there's a membrane protein that greatly accelerates cellular lysis. So this was quite unexpected, has important ramifications for innate immunity, cell biology in general. So I think that's sort of a quick overview of decades of work. But as you can see, the central theme is death and inflammation and their relationships.
Yeah, that's fascinating. It's interesting too that, and it's sort of quite a typical example where domains are called death domains, and then it turns out they're in proteins that don't really deal in death per se.
Absolutely. Yeah.
It reminds me of an example I once came across, and I've written about a little bit. As you might expect, dung beetles normally survive by gaining their nutrition through the consumption of dung.
Yeah.
But a group, I can't remember where they were doing this, it might have been South America, discovered a beetle that was extremely similar in all of its morphological features to every other dung beetle, but it didn't eat dung. And so with examples like that, I formulated what I call the principle of radical evolutionary indifference,
Yeah.
which says that evolution doesn't care about our categories and preconceptions.
Oh, absolutely. Absolutely. That's a great example of that.
Yeah, and it's also, I think, your retelling of your just a few highlights from your career, and I know there are a number of others, illustrates how one question leads to another and another. And it's so often the case that assumptions that have been made get overturned when you look more closely.
Yeah.
So
Yeah. No,
those are
precisely.
beautiful examples. In light of what you've just described, do you have any particular approach to identifying problems or questions you think are worth pursuing?
Again, I was greatly influenced by what I learned at WashU. One was not to enter a field that is crowded, because it's then very difficult to make a dent, and maybe think more unconventionally, against the grain. And I think you have to find out what's your comfort, what do you enjoy, what's your temperament. And I really like what I would call 0 to 1 discoveries, sort of something new, rather than taking something from 1 to 100, which is more consolidation and refinement. But the way I sort of decide on questions is to think about them, but really discuss them very extensively with my colleagues, and discuss them with a view that they play devil's advocate. So we really want to pressure test the idea.
It's sort of wash you recreated.
What you recreated, because that is so important. Because one can be deluded into one's own ideas and thinking that they're the greatest things in sliced bread. But I think just having those discussions and having a cadre of colleagues who are critical, that is important. Once it passes that pressure test, then we will do some exploratory work, and if there is something to it, if it catches traction, then we will focus on it. But I would say that, and we really, more often than not, take a genetic viewpoint. It's either could be a forward genetic screen, it could be a CRISPR screen, but we try to, the foundation is often laid by the genetics in the system.
Do you think it's fair to say that by taking a genetic approach, you're putting up front the question of whether a given phenomenon or a given gene or protein is important in vivo, so that you don't spend time discovering in vitro phenomena that may never translate into what goes on in the living entity?
Yeah, I think so. I think, and I may be wrong here, I just think that the tools of biochemistry are so enormously powerful that one can show a lot of things in a cell-free in vitro system that may have no bearing to the living organism. And so we prefer to do it in reverse. We want to first make sure that it is of critical importance, and then work out the biochemistry.
Is that primarily the result of your sort of personal approach or your personal preferences, or is it something that is more appropriate for your current environment, which is a for-profit company, than, let's say, if you were still at a university?
No, I think I've always felt that. I mean, clearly we change as we with experience, but my experiences have been that the work I've been proudest of and the work that has stood the test of time largely has been work that started off with genetics. And then we built on that, the discovery of, Dermin D, the discovery of NINJ1, the discovery of the non-canonical inflammasome pathway. Those were all very intimately linked to the genetics of or very intimately linked to a genetic interrogation of the pathways.
Do you accept the hypothesis that the immune system responds to non-self, or the competing hypothesis that it responds to so-called danger, which has been very much sort of diffusing into the immunology community now for decades. Everybody talks about danger and danger signals, but I have my own views on this question, but I'm curious how you think about it.
You know, I'd say I've been compelled with the earlier work of Charlie Janeway, who, and from that derived the existence of PAMPs and DAMPs. And these are really, one way to think of them is their means to sound the inflammatory alarm. And that's why they're required as adjuvants in a vaccine. I mean, PAMPs and DAMPs certainly alert the immune system, but DAMPs, the danger-associated molecular patterns, arise from within. So, and I think it gets a bit semantic. So uric acid crystals are a DAMP. Are they really non-self? Well, I mean, that's debatable. You could say, well, uric acid crystals are self because you can have uric acid crystals, their deposition, and not have gout. Or you could say over a certain threshold, they represent non-self. But I think the basic premise is that there is a system to recognize foreign and internal molecules, the so-called PAMPs and DAMPs. And this revs up the innate immune system and is the basis for adjuvant use in vaccines. I mean, that's the sort of way I look at it.
I just will mention that there are vaccines that don't have adjuvants. And so, although Charlie Janeway is famous for what he called his dirty little secret,
yes,
that adjuvants are necessary, they certainly can increase responses a lot and make vaccines more likely to be useful. But there have been successful vaccines that don't use adjuvants. I just think at times, the way the word danger gets used, it ends up being almost a tautology. So that if there's a response, it's called a danger signal.
Yeah.
And I think there are areas in immunology where it's very clear that those kinds of signals clearly matter, but they're not the only ones that matter.
Yes. No, I'd agree. Yeah.
Have you ever looked into complement? You mentioned the alternative pathway.
We haven't really studied it. I mean, except to note that they're both protease cascades with built-in amplification systems, and both are subject to complex regulation, because if they go awry, there can be serious damage. And also that they ultimately, when they run their full course, they compromise membrane integrity. So I think there are those sorts of similarities between these two cascades, but we ourselves have not studied complement activation.
I didn't think you had, but I just thought it's interesting because I was recently at a local inflammasome meeting that was put on here by the groups that work on that topic. And they brought in some, first-rate people from outside. And some of them were talking as if the study of innate immunity began with Charlie Janeway. And I pointed out that the study of complement goes back a century, and that not in any way to diminish the importance of the more recent work on inflammasomes and related systems that you've so beautifully delineated, but to make the point that it is really astonishing how many analogies there are.
Oh, yeah.
In terms of protein-protein interaction leading to protease activation and signals, devolving from there. And, it doesn't replace what work has been going on studying intracellular systems, but it's fascinating that there's an extracellular system that is enormously similar. And as you pointed out, ends up with at least one of the same outcomes, which is the destruction of membrane integrity and the amplification of various inflammatory pathways.
No, absolutely. Evolution never ceases to amaze. I mean, bacteria encode a gasdermin. This was just discovered last year by a group in Boston. And that gasdermin is encoded on an operon that also has a caspase-like protease. So in the presence of phage, the operon is activated, the caspase-like protease cleaves the bacterial gasdermin, which then forms a pore, leading to death of the cell and depriving phage of its replicative niche. So these sorts of cascades that end up in membrane damage seem to be evolutionarily conserved.
That's really interesting. I had not come across that. Wow.
Yeah, there's a paper in Science last year.
I mean, it's interesting to consider that CRISPR itself is a bacterial immune response in a sense.
Yeah, no, it is. And you know, you would say that about restriction enzymes as well. And so, and I say like restriction enzymes and CRISPR are not present in mammalian innate immunity, but certainly the gasdermins, you know, bridge all bacteria to humans in terms of the conservation of the evolutionary principles of pore formation.
Is the bacterial gasdermin-like molecule, is it actually similar in primary structure, or is it just similar functionally?
Oh, it's similar in primary structure. Yeah, it's
So
quite remarkable.
wow, that is quite a finding.
Yeah.
In your career, you said you moved to Genentech in '97, is that right?
Yes, yes.
So have you done only basic research since you moved there, or have you had more product-oriented roles while you've been there?
Both actually. So besides running my research lab, I also serve on various committees. Probably the most prominent one is the Research Review Committee that oversees research at Genentech and oversees the early pipeline. We meet twice a week. And it really gives me a bird's eye view of drug development. And at the end of the day, the realization to get a successful drug is nothing short of a miracle, because there are just so many, you know, slips between the cup and the lip. I mean, it's just amazing. But it's a committee I've worked on for a long time. It's one that I learn a lot from. It's a lot of fun. The other responsibilities that I had, the postdoc program at Genentech. We have about 140 postdocs on campus, and that's just a really enjoyable job because postdocs at Genentech aren't allowed to work on a product-related project. They have to do basic science, fundamental work. So that's, you know, it's great to have that environment within a company.
Yeah, that's interesting. It's also, I wonder, is that unusual? I mean, would most companies limit postdocs totally to basic science? I would guess not, but you probably know.
Yeah, I think it's unusual at this scale that we have such a large program devoted to it. And it's been in existence for a long time, for 40 years. And so, yeah, I would say it's quite unusual to have a program like this. Of course, in sort of more mature industries, in the physics industry, or in the physics-based industries, IBM, Bell Labs, et cetera, there has been a strong tradition of basic research.
But in terms of biotech companies, other biotech companies.
Yeah. I think fortunately, the good news is that there are companies that are moving in that direction. Novartis, Regeneron come to mind, who have postdoc programs. I think companies are seeing the value of investing in basic research, and I hope that trajectory continues.
That's a very interesting development, because if the companies recognize the value of the basic science, that's quite impressive, actually. What are your thoughts about the interplay between basic and more practically focused or mission-focused biomedical investigations?
There's an overemphasis in academia and granting agencies on translational research. I really think that these organizations should be fostering basic fundamental work. You know, we just discussed restriction enzymes and CRISPR, and there are other discoveries that were just curiosity driven. And I increasingly get the sense that granting agencies are configured like they're funding engineering projects. They use words like timelines and deliverables. And I don't think that's the best investment of research dollars. And I hope the pendulum begins to swing the other way, where there is greater emphasis on curiosity-driven work.
I expect that people will be taking notice that someone who works at a company believes that academic and granting agencies should focus on basic science and not pretend that they're all about application. Not that, not that there's anything wrong with that. It's just that in the long run, I assume what you're arguing is that investing in the basic science will probably pay off more completely.
Oh, and that is key. And, the pharmaceutical biotech industry is built on the foundation of the work done by the NIH, by basic science. So, I think each should do what they do best. And I think the universities shouldn't steer into translational research too deeply at the cost of basic research, because that's where industry is reliant upon the basic research done at universities.
Yeah. Although Genentech is unusual in that you have...
It's unusual, and that's good, but it'll never be a substitute for,
as impressive as it is for a company to have that large a postdoc program and as much basic research as you have there. It's not, in terms of scale, close to Harvard Med School or NIH. So, but it's a very interesting perspective. Have any of your specific discoveries yet been transformed into therapeutic or diagnostic applications?
There are two programs there we did a lot of the foundational work on. There's a kinase, RIP kinase 1, that is a key kinase in the necroptotic pathway. And then there is a sensor, NLRP3, in the inflammasome pathway that mediates pyroptosis. Inhibitors to both those programs are in the clinic by Genentech, Roche, and other pharma companies. So we'll wait and see what transpires, but that has been satisfying to see work progress into the clinic from sort of the bench side.
What are your views on, or your thoughts on scientific publishing as it is today, which is probably quite different from when we started?
Yeah, I think it's broken. And I think few would argue about that. I think when you look at the history of publishing, it was created to serve a very small community of researchers. It's evident for people who have read The Eighth Day of Creation, The Birth of Molecular Biology. If you haven't read it, I would urge your audience to read it. It's a great book. But you can appreciate that it was a very small community of researchers. They communicated by letters and meetings. But today we have a massive academic industrial complex that spews out thousands of manuscripts a week. So, it's complete exponential growth. I don't have a ready solution, because the problem is of such great scale, orders of magnitude scale in such a short time. But I think it's imperative that the community come up with an alternative that satisfies most people. That's a workable model.
Are you in favor of online publication of manuscripts as preprints prior to peer review?
Oh, absolutely. I think it disseminates the research faster. And in many cases, that work is funded by the taxpayer. So I think it's imperative that it be disseminated to the community.
I guess I'm talking about entities like bioRxiv and medRxiv. But, even if they go up before they have peer review, they attract comments once they go up. So it's not without any, commentary.
It is not without any commentary. Now, I have to say, we ourselves have not indulged in that, but I think it's more a reflection of sort of old world thinking on our part. But, I see bioRxiv papers all the time, and I appreciate them. I just think that claims could be made in such manuscripts that are incomplete
Right.
And since they are part of the record, it becomes correction is always a difficult task.
Yeah. Well, as with most human activities, there are pluses and minuses.
Minuses, yeah.
So, do you have any preference about anonymous versus open peer review?
I think it's another one struggles with. You can see pluses and minuses. Again, with anonymity comes the danger of, completely unreasonable reviews.
Yeah.
And I think you see the danger of anonymity in social media. I think with the open system comes the danger of earning cookie points. You say, and people will review a paper and be known as the reviewer, and then there may be a quid pro quo in the offing it at a future time.
Right.
Again, I don't have an answer to that, but I've seen it work both ways.
I mean, as you're speaking, I'm thinking the problem with both is that either one relies on good faith, and there's no way to guarantee good faith 100% of the time and 100% of the people.
It's a human activity and has all the follies of human activities. Exactly. And I think editors play a very important role in this, but they are also really overwhelmed with the number of manuscripts they handle, and they are handling manuscripts in very diverse areas. So it's, I think, almost an impossible task for them to ride herd over reviews that are unreasonable.
As an editor, yeah, it can be very challenging. I don't know if you know this, but at Pathogens and Immunity, we review articles submitted in any standard format. And we only require our particular publishing format after a submission is accepted for publication. Would you endorse that sort of approach for journals more widely?
Totally. It's a common sense. And you know what? It would save tens of millions of dollars in wasted effort and much anguish.
So as a final couple of questions, let's move to guidance for those who wish to go into science. Do you have any particular suggestions about what they need to know to go into a career in biomedical research?
A career in biomedical research can be very fulfilling. There is the thrill of discovery. There's really, for many people, no substitute. But it's imperative that one be absolutely passionate about it. And if one is not, it's not worth devoting one's time to a biomedical career, because there are many bumps in the road. There's much adversity to overcome. And so if you're passionate and you enjoy the thrill of discovery, you'll be more accepting of those bumps and adversity. But if you're not absolutely thrilled with discovery, you'll get frustrated. So I would say that people need to ask themselves, and there's no right answer or wrong answer, there's no good or bad, is that, does it really drive them? And if it drives them, that's great. But if it doesn't, then think of something else.
I've been devoted to medicine and science for many, many decades. And nevertheless, when I started hearing this term STEM and this constant drumbeat about focusing on science, technology, engineering, and math, all of which I think are great, but I don't see any reason to exclude other fields. And I'm wondering how you look at that concept and that kind of promotion versus promoting a broader standard sort of liberal arts kind of approach to people. Again, I don't think there's a one size fits all formula, but in general, would you encourage people to focus on STEM subjects if they want a career in science or any of the related areas? Or would you counsel a broader range of academic and even non-academic experiences?
I'm convinced that broader is best. I favor a liberal arts education. I mean, I would actually do away with most AP classes. I'm actually quite sure that many students who do AP classes don't develop an in-depth understanding of the complex material that's presented to them, but rather pass the test through a combination of rote learning and exercises. And so I may be a bit extreme on this, but I think a liberal arts education, doing away with AP classes, I think that's the best way to go.
Well, that's a wonderful place to end, and it's been a true thrill to have this conversation. I wish we could have had this conversation periodically for the last 40 years.
Well, we think alike, I think, on many issues. It's been great, Neil.
That's all it is. But I also have to say you're extremely articulate in expressing your views, and they reflect a great deal of thought, which is inspiring, quite frankly. So thank you again, Vishva.